Okay, so we're live. Hey everyone, welcome
Hey,
for joining.
everybody.
Thank you for for joining us. I Tomas.
Hi. Hi, Adam.
Let's give it about. I see people are starting to watch us, but I'm going to give it about 1 to 2 minutes, or people won't miss the introduction, and then we'll get started
Okay, perfect.
officially. Tomas, remind me what city are you in right now?
Sorry, the city?
Yeah, remind me what city you're in.
So I'm currently living in Strasbourg, actually in France, at the border of Germany, and I'm so partially living also in Heidelberg because I'm working at the European Molecular Biology Laboratory, located in Heidelberg at in Germany.
How far is Heidelberg from Strasbourg?
It's 150 kilometers far from Strasbourg.
So do you
So
usually
it's,
drive there or you take the train?
yeah, usually I drive there. Yeah, yeah.
Yeah, so it's about two hours, 2 hours and a half.
Yeah, a little bit less. Yeah, 1 hour 30, something like this. Yeah,
Nice. Okay. And you go there every week or only when you
yeah,
need to?
yeah, absolutely. So I was there yesterday as well. So usually I go 3 days a week on site, and two days remote. So
So that's where you have all your meetings. You go to Heidelberg to have all your
yeah,
meetings.
exactly. Yeah, yeah. I'm trying to optimize my schedule to pack all the meetings on the same on the days when I'm on site actually. So
Oh, better. Yeah. Yeah, because it's a lot better to have meetings like in person than virtually. It's
yeah, exactly. Yeah.
okay. So there's 2 minutes, so let's get started. And as people join us, they can ask any questions, and they could hear you as they go. Let me see. Let me bring up the my list. Oh, I forgot that I closed it. Um, Closed on me before when we were preparing. So let me bring it up right now. Okay, Tomas, our hangout of the month. This is our hangout of October. All right. So here we go. So welcome everybody to the Plotly Hangout. These hangouts, as a reminder, are a space where you can meet and interact with data scientists, data analysts, and engineers merged in the world of data visualization, Plotly, and Dash. You will often hear from our guest speakers about their professional experience, the journey that brought them to where they are today, and the lessons that they learned along the way. Now, your participation in the Hangout is highly encouraged. The more you participate, the richer the Hangout will be. So feel free to unmute yourself if you join us here on StreamYard, or join us on camera, or ask our speaker any question that you would feel comfortable being asked yourself. So people who are joining us and watching us from our social media channels, you can just ask a question and I'll receive it here in the comments, and then I'll relay them to Tomas to answer. Um, and if you are more of a chat person and on with us here on StreamYard, you can use the private chat as well. All right, so to get started, I'd like to introduce Tomas Weber, a research fellow at the Data Science Center of European Molecular Biology Laboratory in Heidelberg, Germany, and he's part of the ARISE program. He leads the development of Depictio, a platform that enables researchers to create and share interactive dashboards and web applications directly from bioinformatics pipeline outputs using Dash. We'll talk about Depictio today, but as a summary, Depictio leverages Dash's flexibility to deliver shareable, interactive tools that help scientists explore, interpret, and communicate the results with ease. Now, during the Hangout today, we're going to cover a few things, mainly throughout and using the Depicto app. But you're going to hear all about turning complex bioinformatics pipeline outputs into interactive Dash apps, the importance of keeping part of the system close to the data, and scaling Depicto from individual projects to institutional use with Kubernetes-ready setup, built for reliable production-grade deployment. So, Tomas, you're going to teach us a lot today. You're going to talk about a lot. I'm excited. Thank you for joining us.
Yeah, thank you a lot for having me today and giving me the opportunity to talk a little bit about the project as well. Thanks, Adam.
Of course. So before we go into the app itself and dive into Plotly and Dash and how you use it, Can you give us a little bit more of a background about yourself? Like what did you study?
Yeah, so I started actually studying environmental engineering in 2012 with a bachelor's actually in Strasbourg, the city I'm still living in, and so I completed actually that degree, but then I reoriented myself and discovered bioinformatics. So where I continued with a master's degree actually afterwards, still in Strasbourg. And during that master's, during my master's thesis and my internship, I had the opportunity to continue with a PhD and a fellowship actually to evaluate and assess pathogenicity of mutations and their link to rare diseases in human using machine learning. And so that's how I got into data science actually, and into Plotly and Dash, and all the technological stack that helps researchers to better analyze their data actually. And so now, since March 2022, I'm working at the European Molecular Biology Laboratory in Heidelberg as a research fellow in the context of the ARISE program, which really focus on research development actually to support researchers by developing tools to accelerate and facilitate their research.
A lot. You did a lot of studying, so that's pretty cool, and a very interesting thesis as well. For our maybe younger audience, Tomas, that are watching us and are thinking about the field, and studying bioinformatics, and let's say they come to you and ask you, Tomas, why do you think what interests you in bioinformatics? Like, what's cool about it? Why should I do it? Obviously, it depends on personality. But in your opinion, given that you've been in the field for a while, what would you suggest to somebody that's thinking about studying it?
So I think so. At least in my experience, when I started learning about bioinformatics, so I always was fascinated as a child by computers. I was always fixing them and so on, but I didn't really know a lot about programming. And then I started obviously my studies with a focus on biology. And when I started studying programming and bioinformatics, it really scaled up the potential and the ability to actually apply your analysis on thousands or millions of sequences with batch processing, access to remote databases, those kind of things. And I think that bioinformatics, and now also with the rise of AI, is really a cool topic, because there's still a lot of potential and plenty of things to do. And there are big publications, Nobel Prizes recently with AlphaFold, for instance, that is directly related to the field. So I would say, there are still almost everything to discover with that research area.
And would you say, Tomas, that in order to get a job in the field, like a good job in the field of bioinformatics, is it absolutely necessary to have a PhD, or people can also get a job with a master's degree?
I would say it's definitely possible to have a nice job with a master's degree. Now, more and more research institutes, and especially and companies as well, are most of the time looking for PhD, but it's still possible to have a master's and actually accumulate experience over time, especially on many different topics. And also, it's not because you have just a PhD that you can do everything. You can have a master's and be super and be excellent with your programmatic skills or other things that you can bring in data science. So definitely with a master's, it's possible to start your career in bioinformatics. Yes.
Nice. Okay. And then you mentioned that you started you discovered Dash and Plotly throughout your studies. What was that, your bachelor's, or during your master's you discovered Plotly, and what did you discover first?
So actually, it was mainly during my PhD, but during my master's already, I was comparing bioinformatics softwares that are actually specific to identify structural variations in the human genome. So large events that can be duplication, so basically a copy or deletion, those kind of things. And I built a benchmark for this. And in my thesis, actually, in my PhD afterwards, I really leveraged a lot Plotly and Dash, because I needed actually exploratory tools in data science to compare many different variables, and building basically databases, and have the possibility to query that database in many different orientations. So that's how I got into Plotly and Dash, and because of all the interactivity and the possibility to share with colleagues as well, with my supervisors and so on, that was really a benefit for me to rely on Plotly and Dash. Yes.
Nice. And so you have a few years of experience with Plotly and Dash. Would you say, which is kind of cool to see how you've seen how Dash developed over time. Um, you'll show us this in the app with more detail, but do you also use any community built components and libraries that sit on top of Dash?
Yes, absolutely. So as you will see in Depictio, I'm trying to stay up to date with all the community developments brought by many fantastic people that especially are listing their work on the forum, which I definitely recommend to consult on the Plotly forum. And so I'm using, for instance, a grid Dash. Dynamic grid layout, which allows to drag and drop components on your app and resize them as well. I'm also using Dash Mountain a lot,
Okay.
which is super powerful and helpful to improve the styling and also the client-side properties of your application. I can't list all the extensions from the top of my head, but Dash
It's a lot.
extensions is, yeah, it's a lot available. Dash extensions is nice as well. Dash Flash. So yeah, many
Dash
Dash.
extension is really powerful.
Yeah,
Like
yeah. Dash AG grid as well is super nice and powerful. So
cool. All right. Well, you got me excited now talking about all the components you use. Do you want to share your screen
yes,
and,
absolutely.
and talk about this app and how it came to be? Like, what was the reason you decided to like create this app?
Yeah. So I will start sharing my screen. Share.
I forgot to tell you this at the beginning, Tomas. Just make sure, because we're sharing screen, to like turn off any notifications you might get or
Yeah. So I try to enable the do not disturb mode on my Mac, so
good.
that should be fine. Can you see it now?
I can see. Wait 1 second. Let me upload it to the stage so everybody can see it. 1 second. Okay, we all see it.
Okay, great. So actually, maybe before getting into Depictio itself, I wanted to give just a 30-second summary on bioinformatics and bioinformatics pipelines. There are plenty of technologies available, but I will just have a focus on this technology called Nextflow and NFCore. And basically, there is a nice community. So similarly to the Plotly Dash community, other type of communities are building around other technologies. And here, people were working to develop and agree on how to define a workflow, basically combining a set of tools to conduct an analysis on a specific type of data. And I will present actually an app that allows to aggregate the results at the end of those workflows to help researchers build their web application at the end.
Perfect.
So that's just what I wanted to show you first. And so basically, this is my system actually, where when you land on the system, you can already visualize the different Dash application, web application and dashboard that you built with integrated thumbnails that are generated on the fly when you save the dashboard. When you are not owning the dashboard, you can duplicate it and become you can create a copy of it actually to become the owner of your own version of the dashboard. And so I can show you an example, but whenever you go on an app, then you have the possibility to play with all the interactive components to, let's say here, so here it's just the iris dataset as an example. But whenever you modify things, it gets updated in real time and so on. And so here you don't have the possibility actually to modify the dashboard at all. You cannot edit it. you can only play in a stateless manner with the interactive components.
Like a user.
Like a user, like only a viewer to share with colleagues, with end users, people that would like to consult your data set and explore the end visualizations you generated to see how you generated your insights and the final analysis in your research, actually.
Now, Thomas, this is, I forgot to ask, this is that people using this are your research colleagues, or who exactly?
Yeah, so it's my research colleagues mainly at the moment. So I have two main big use cases at the moment, but the platform was also deployed in SciLife Lab, so the Swedish Institute for Life Sciences that has like a Kubernetes platform to help researchers host their Dash application, Shiny application, Gradio, and so on. And so the system is available also as a standalone tool to basically allow researchers to register data and build their application. But at the moment, I have two main use cases, and I will present just one of them after the Iris dashboard. And I just wanted to show that once you land in your own version of the dashboard afterwards, so here you have the possibility now actually to lay out and modify and change the result of your dashboard the way you want. So here something happened. I don't know what it's the beauty of a live demo, but yeah. So basically, you have the possibility to resize components and so on the fly, and everything is responsive horizontally and vertically as well.
Nice.
And so it's possible to disable that edit mode, but also to create new components actually. So here, when you add a new component, you can pick the type of component you want to add on your dashboard. It can be a figure, card, matrix card, interactive table, some text to lay out and arrange differently your dashboard, or also multi-QC component. I will come back on this later. And so let's say we want to build a new figure, still using the Iris dashboard. So you can actually access all the different type of Plotly Express functions that are registered in the system. So you can build a histogram, bar plot, box plot, scatter plot, and so on. And whenever you create your visualization and pick some functionalities and options here, it will update in real time and generate the figure for you to actually render your component and add it on the dashboard afterwards. And so you
get to see your data in the graph before you save it, so you understand if this is what you want or you want something else.
Yeah, exactly. Yeah, and it's also possible to edit it afterwards.
Okay.
And so there is also a code mode which is still in beta, running with restricted Python actually, that I only allow to execute polars commands and Plotly Express command at the moment. And I just wanted to show you an example, let's say with a bar plot where so it's the functionality here, and the improvement is that you can have a first pre-processing step to actually generate a temporary data frame based on your raw data. And so here, for instance, we are aggregating some columns to compute the mean and the standard deviation, and some columns in the Iris dataset to build that bar plot with the error bars here on the y-axis.
Nice.
So afterwards, you can save it, add it on your dashboard, and the newly created components. So just take 2 seconds to be generated, and then you can arrange and lay out it on your dashboard, and it becomes directly available and responsive to the rest of your functionalities.
But Tomas, what was the reason you added that code? One of the reasons that you added that code feature in beta version is that also people who, because right now I'm looking at your app, and what's cool about it is that, and so helpful, is that you don't need to know how to code to create your graphs, right? So
yeah.
But you also created that code part so people who do know the code and want to use code can actually use it as well.
Yeah, so that was two reasons, actually, two main reasons. The first one is to allow people that already have like Jupyter notebooks or other type of analysis to reuse figures that they already generated to be scaled at the production level and aggregate results over time, actually at a different scale. So that's the first reason. And the second one is to have this possibility to actually have a pre-processing step for the data frame to be able to create another view on the data frame and not rely only on the raw data frame. But let's say if you want to group by or compute some average on something and display it differently, like here you could not, with the default Plotly Express functionalities and the raw data frame, compute the error bars like this.
Okay.
So that's why I wanted to have this additional mode, actually.
Okay.
So this is an example with the Iris dashboard, and I will show here another example, actually, that with a demo dataset. So it's not actually production data that was not already published, but this is actually leveraging the pipeline I just presented, the NF Core pipeline, focusing on microbiome analysis from different type of habitats. So including groundwater, river water, sediment, and soil, and where we are interested into analyzing the diversity of species in those samples, actually using different visualization. And so here you have, for instance, the taxonomic composition by sample. Samples are represented here in the x-axis, and you have basically everything which is scaled in percentages. And you have different quality control metrics here and measures that are generated using the MultiQC tool, which is a super nice tool. And actually, this is why I'm leveraging those pipelines as well, because this software MultiQC is available in the end of every pipeline that the community agreed on. And this tool is actually generating Plotly Express plots on the fly for each sample, and I'm aggregating all those data and plots to build a single plot which is responsive to filters, drop downs, and so on.
That's a lot of information that you're processing.
Yeah, actually, yes. And so this is why sometimes the system is still a bit could be improved in terms of performance. So I identified already some points to improve the efficiency and the performance on the system. But there is a lot of things happening under the hood, actually. And so here it's possible, for instance, to filter by habitat type, and automatically all the figures are responsive in real time, and you have informations at different levels. You can also filter by collection dates, by type of samples, by phylogenetic diversity, for instance, or taxonomic classification. So it's possible actually to leverage that dashboard in different ways, and you can see that there are different type of components available on this dashboard.
I like also the summary cars in the top right.
Yeah, so actually it's possible as well to build those summary cards with the menu I presented before here.
Like a new feature, a card? Oh, a card. Okay. And these are automatically summary cards?
So you just need to pick actually the column you want from the data frame you want to generate the card on. And let's say it's a numerical column like the width of sepal here. You will automatically have access to a range of aggregation methods like the count, sum, average, median, min, max, and automatically you have the value computed, and you can change the icon, the color, and so on, actually. So that's
Wow.
that's how I define the system. And so then it gets available here, and so you have the trend line as well when you change some parameters. So
nice, very nice.
Sorry. So yes, these are two example dashboards. You also have access on the system to a menu to take some notes and documentation using the rich text editor from Dashmantine, actually, that you can put full screen as well.
Dashmantine text editor, really cool.
Yeah, this is the rich text editor from Dashmantine. Yeah.
Wow. So do you know if your colleagues use that a lot?
Yes, a little bit, not a lot, I would say, but to take some, for instance, if they notice something interesting on a specific, by doing some filtering on a specific combination of things, they can take some notes here at the bottom, or also to copy and paste some example code they would like to incorporate in the dashboard afterwards using the code mode beta. So these are kind of features that they are interested to use. Yeah.
Wow, that's so many. How long did it take you to build this dashboard?
So the dashboard itself not that long, actually, using the graphical tool that I developed, but the platform itself quite long, to be honest. So I'm working
why did the platform take more time?
Because the platform is quite complex. Maybe I can show you. Sorry, I didn't have it open.
That's
I wanted just to show you one slide, not a full PowerPoint, but just one slide here.
Sure.
About the architecture of the system. And as you can see here, the architecture is quite complex.
Oh, but wait, right now we're only seeing.
Yeah, sorry. Yeah, I know why. Okay, just give me 1 second. Stop screen. I will share again. Yeah, that should be fine again now. Can you see it?
1 second. Yeah, so let me add it to stage. Okay, we can all see it.
Yeah, thanks. Sorry. So yeah, the platform is quite complex actually, because there are some metadata processing in the backend with some caching, no SQL database. Everything is asynchronous here using FastAPI, MongoDB, and so on. And then there are some external connection to S3 buckets to store and accumulate the workflow outputs over time. And then I started to work with asynchronous callbacks as well on Plotly Dash, but most of the callbacks are still synchronous at the moment. But the platform is quite complex, so that's why it took me some time to build the system actually.
Okay. Now, you did mention that you wanted Tomas to maybe make the system a little bit more, even more efficient, even more faster, because as data accumulates, it might be a lot. What were your thoughts? What did you have in mind?
So actually, at the moment, I'm using. I can show just an example here. I'm using that system called Delta Lake as a format to save and store all the data that was aggregated into a single small data lake file, actually, which is built on top of Apache Parquet, which is already super efficient. But the system is already using pattern matching callbacks, meaning that each component has its own callback to refresh and render the value of the card, the data inside the plot, and so on. But there are still some round trips where there are some contacts to the API first that are going back and forth, and I would like to optimize those aspects to have something a bit more efficient and a bit more stable in the coming weeks and months. So I identified some of those points actually already, and alongside other aspects and developing more features and more compatibility with other data types, I would like to work on those. Yeah.
What about there's pretty advanced features that you can do with the Dash callback. Are you planning to use any of those advanced features like patch or the matching callback?
So yes, actually, I'm already using pattern matching callbacks for all of those. So when you refresh something, it's directly triggering in parallel those callbacks to render the different components individually and in parallel. I would like to leverage as well the background callbacks using a Celery worker to offload actually the processing when the data gets really large, actually, to when you have more than 1 million rows, it could be cool to offload your processing to a dedicated worker doing just the processing for you and giving you back what you want to observe and visualize. So this is something I want to work on as well. And also work maybe to at the moment here, I don't know, so it's not directly visible, but I'm using different data frames that are joined dynamically. So there is not like a single data frame, but rather different types of data that are interacting with each other. And I would like maybe to rely more on DuckDB as well to leverage the efficiency of the joins available in the technology. to improve the efficiency of the system.
Are you using polars or pandas under the hood?
So polars, polars everywhere. Yeah.
Did you find that it made it faster?
Yeah, absolutely. Especially when it comes to the first aggregation step, where using this is what I wrote as well on the description for this Hangout today. But I built actually a CLI tool, which role is to aggregate and scan files in a single Delta Lake. And
Okay.
this system is using polars, and you definitely see an improvement using polars over pandas when you aggregate a lot of files and leverage all the lazy operation that polars integrates. Yes.
Cool. Now you had a lot of, this is a very informative dashboard. Let's go over just one or two of the polygraphs because some of
Yeah,
these
sure.
would be cool. Let's go over that. Is that a heat map on the top right?
So yes, actually, this is a heat map showing actually different quality measures with three different levels actually. So green is usable, yellow is okay but borderline, and red is not really passing the threshold regarding that specific measure. And on the rows here, you have all the samples processed, and so you can see that. For instance, all those samples are not passing some metrics that are usually used in bioinformatics when it comes to the sequencing. So, for instance, people are usually comparing the amount of GC content in the sequences. So you may know that DNA is always composed of 4 different bases, A, T, C, and G.
Okay.
And those we know the measures and the ratios of AT against GC, and this is good information when the sequencing is going wrong actually. And so this is a kind of measure that people can rely on to know if samples are usable or not. But as I mentioned at the beginning, this is an example demo dataset that was not published. So here it was just for the sake of showcasing actually, but this is not real data actually.
Right, right, just for the showcase. Okay,
Yeah.
and then if you can scroll down a little bit.
Yes.
So what these are, that chart at the bottom, is that a cumulative chart? Is it a line chart? How did you build that?
Yeah, so this is an alpha rarefaction curve actually to see how the population or what is diversity of species available in a sample
Okay.
according the sequencing depth. And so the more diversity you have, the more you have different species present in your sample. And so for every sequencing depth, if you zoom, you see that you have actually error bars for
Wow.
every point. So it
Nice.
can be quite informative. And so if you filter, let's say, for a specific habitat, you will have actually the same figure, but only for three samples, and you can compare maybe between a sample from that habitat with another to check what is diversity according that specific measure actually.
Really cool. So your colleagues already in your research institute have already been using this, and based on their feedback that you get, do you change or modify things? Like, are you also thinking, for example, of adding other graphs?
Yes, actually, yes. So this is why the system is cool, because they can actually leverage my what I'm calling a template, where I build this template for them, but they can also take the ownership of that dashboard using the system I presented at the beginning here, and create their own version of the dashboard if they want to add a new figure or modify one. So this is completely feasible and possible. So this is why the system is cool for them as well, and to also combine not only those more, I would say, generic type of visualization using Plotly Express, but also this kind of visualization that are more complex to generate, I would say, using Plotly Express, because you need more steps in the code. So using multi-QC, it's really cool to be able to leverage and aggregate data over time, and to suggest kind of one dashboard integrating different levels of analysis.
But Tomas, you're obviously an expert in this because not only because you built this, but because you're an expert in the field and you know to work with Quality Express very well. You know what you need to put on the x-axis, y-axis, color, legend. You know all that language that maybe some of your colleagues don't. Does that mean that because of that, some of your colleagues will? Do you find that they actually use the feature to copy and then enhance a different template, or they would just come back to you and say, Tomas, can you build this graph for me? This is really cool.
I would say it really depends on the people. Some of them are more like focused on the wet lab part, and so they are less familiar, as you said, on the coding and programmatic topic and part. So they will rather come and have a feedback to modify and enhance something. And some others are already experienced in bioinformatics and in programming, and they will know the features of Plotly Express, so they will directly know what kind of variable to modify or how to improve a figure themselves, I would say.
Oh, okay. And another question I had is, do you have always more or less the same amount of colleagues and people using this, or do you expect it to really grow and other people from other institutions to use this, which will then obviously introduce another challenge of how do you get so many people to use it at the same time?
Yeah, actually, so this is a really good question. And so at the moment, it's still mainly used by so internally with different use cases parts that are part of the laboratory. I'm trying to build on new projects with similar data, actually, leveraging new components, including a geograph component to display a map. So that will be one of the new use cases, but it will still be internal. And when it comes to giving access to other institutes and so on, so I'm providing a guide to host for the institution and people to host the system themselves using either Docker Compose or Kubernetes. And so I already have a Helm chart available to install all the complete system on Kubernetes, including replicas and so on for Dash and for FastAPI, actually. And so for Swedish scientists, it's possible to directly log in on one of the SciLife Lab platform and leverage actually the implementation of Depictio on a system called Serve, and they can directly access Depictio there as a standalone app like RStudio, Jupyter Lab, or VS Code. Yeah.
Wow, this and this app, from what you're telling us, it seems like this app is so helpful and contributing to the researchers' work and people in the field that they will probably depend on this to really do even a better job and understand their data more. Does that mean that this app will you continue developing it as long as you're in this position?
Yes, absolutely. I would like to continue developing it with to improve the performance and also the set of functionalities available, and to develop also templates for the community, similar to the template I developed for that specific dashboard, which is quite heavily used actually by the community, but also to other type of analysis in the sequencing world. So I would like to focus on this. If possible in the next months. But obviously there will be some challenges as well to get people on board and help them to integrate their data. So this is why I'm doing some rounds of improvements and so on using the different methodologies here.
And you must love coding in Dash and building these dashboards.
Yes, absolutely.
Like, why go to meetings? Why do anything else? Just spend 100% of your time doing this.
Why not?
Good. All right, Thomas. Thank you so much for the presentation. This was very informative. I really appreciate it. We will add the. Let me unshare 1 second.
Yeah, thanks.
We will add the link. If that's okay by you, add the link to the app under the video description.
Yes, absolutely. Yeah, everything I presented today is actually publicly available in a demo running on Kubernetes, on the Kubernetes cluster of my institute. So feel free to give it a try and provide feedback. And there is also a link to the documentation. Everything is open source and publicly funded by the European Commission. So,
Perfect. And don't forget to send me if you want me to add any other links and under the recording, under the video description, like your LinkedIn profile, anything else, just send it to me after we're done. Okay.
okay, great.
One last question, Thomas. One last thing I like to ask our speakers, our guests, is what is one thing? Let me actually ask it the same way I always ask it. So one concrete suggestion or advice for our viewers. What advice would you like to share with our 70-something people watching us today? And it could be anything, Thomas. It could be about Dash. It could be about building. It could be about the field of bioinformatics.
So actually, on Monday I was in a very nice symposium, really cool thing combining Earth observation, so satellite data, and molecular biology data. So it's super really the two opposites in terms of scales. And there was one speaker, I think from NASA, which was presenting, and at the end of the talk saying, keep things always simple as possible. And so that will be my advice, even for myself. After more than 2 years developing this, I'm realizing that I developed some features that are too complex for people and even for myself. And just keep things as simple as possible to start with, and don't overdo and over complexify when it's not necessary. So that would be my advice when you're starting programming, especially. There are many and a huge amount of things and technologies available. And keep it simple first and efficient, and you have plenty of technologies that are cool, including Dash, that are super efficient, and don't overload them and overcomplexify.
Wow, that's great advice. All right, Tomas, thank you so much. I really appreciate your presentation. Everybody here watching us, don't forget we have our Figure Friday sessions every Friday at noon Eastern time to talk about the data set of the week and the visualizations of the community built. We actually have a very cool Dash app that was built by Alex, one of our community members, on Halloween trick-or-treating. So that's going to be interesting. And then tomorrow we have our virtual Plotly community meetup. For anybody around the world who wants to join us, just send me a request via email or LinkedIn. You can find me, and I will add you to the calendar. It's tomorrow, Thursday at 1 p.m. Eastern time. We'll have several people, including Tomas, that will have a few minutes to speed talks to showcase their apps and their creations. And you'll get some Plotly updates. Just email me at adam@plotly.com or adam@plotly.com. And I think that's it. Tomas, thank you again. Thank everybody for joining us, and have a wonderful rest of the day.
Thanks a lot, Adam. Thanks everyone for joining and attending the meeting.
Thank you, Tomas.